Welcome to another series, MCQ series. Let's continue our questions on enzymes. Enzymes are mostly catalytic proteins. The answer is catalytic proteins. Catalytic proteins. They are catalytic proteins. So they catalyze biochemical reactions. They catalyze biochemical reactions. They are catalytic proteins. Myosin is responsible for muscle contraction. So myosin has there are two components: the actin and the myosin. There are two muscles that help. They are proteins actually. They are proteins that help in muscle contraction and relaxation. Actin and myosin. Okay, you can see actin and myosin interaction causes what? Muscle contraction and relaxation. Now, protease catalyze protein hydrolysis. Protein hydrolysis. Albumin transport dash hormones, drugs, fatty acids. So the answer is all of the above. Which level determines the overall 3D shape? Tertiary. Tertiary structural level determines the overall 3D shape, which is called the three-dimensional shape of a protein. For quaternary is when two or more polypeptide or two or more tertiary proteins are connected together, but not by covalent linkage. Not by covalent linkage. They are attached to each other, but not by covalent bonding. That's why we have the quaternary structure. The secondary structure, we have the beta sheet and the alpha helix, which are stabilized by hydrogen bonding. But tertiary structure is stabilized more. There are other stability factors. Example, ionic interaction between amino acid side chains. For example, if we have glutamic acid, which is acidic in nature, that has the COO minus, and we have lysine that has a positively charged arrow group. So you can see negative and positive can interact, causing more stability to the protein. We can also have hydrophobic interaction. Hydrophobic interaction are usually seen when the amino acids are non-polar. When the amino acids are non-polar, so they form hydrophobic interaction, which can also promote stability of the tertiary structure of the protein. The denaturation that may cause dash, may be caused by dash, extreme heat and extreme pH. Of course, extreme pH can denature a protein. But when we talk about extreme pH in protein denaturation, for example, if we have trypsin or pepsin, which are proteins or enzymes that act on extreme low pH, like 1 to 2, you can say yes, that particular pH, they will have their optimum activity. So every protein have its own optimum activity. So for one protein, that particular pH might be okay for the protein activity, but might not be okay for another protein or enzyme activity. Now, which protein is a hormone? Insulin. Collagen is a connective protein. Keratin is a fibrous protein. Actin is a protein that is usually required for muscle contraction and relaxation. SD8 page separates protein mainly by dash. When we say SDS means sodium dodecyl sulfate, while the page means polyacrylamide gel electrophoresis. So when we use SDS page, we separate protein based on size. Separate protein based on size. Now, pepsin work best at dash. Neutral pH, acidic pH is called acidic pH. Why acidic pH? Because pepsin is an enzyme that is found in the stomach. and the stomach pH is acidic. And trypsin is secreted by pancreas. Secreted by the pancreas. Structural protein of hair is keratin. Keratin forms the structural protein that is found in hair. Protein folding is assisted by chaperones. And that's chaperones. Now, when we talk about protein folding, we also, like I said earlier, there are several factors that determine the protein folding. One, the hydrophobic interaction between non-polar amino acids, which is a fibrous protein. Collagen is a fibrous protein. It is used for when strength is involved, when strength matters. Talk about collagen. The isoelectric point depends on amino acid composition. So it is the amino acid component of a protein that determines the isoelectric point. So if there are more charged amino acid, either positive charge amino acid or negative charge amino acid, it will determine the isoelectric point of that protein. Which reagent precipitate protein strongly? Ethanol trichloroacetic acid precipitate proteins better. So do not forget to subscribe. Click on the notification bell for more videos on biochemistry MCQs. Do not also forget to follow my videos on amino acid, peptides, and proteins to learn more about proteins, amino acid. You will learn classes of amino acid, essential and non-essential amino acids, the chemistry of amino acids, the classification of amino acid, whether they are hydrophobic amino acid, whether they are hydrophilic amino acid, whether they are polar, whether they are non-polar, whether they are negatively charged, positively charged, or neutral, whether they contain hydroxy group, sulfur group, and the rest. You also learn about protein isolation and purification, where we talk about the denaturation of proteins, cell lysis, the steps involved in protein isolation and purification. So don't forget to subscribe and click on the notification bell